I'm Jim Churnside. I'm with the National Oceanic and Atmospheric Administration. I'm here in East Sound, Washington, studying thin layers of plankton in the Sound. I'm working with groups from the University of Rhode Island and the Naval Research Laboratory. I'm using a Lidar system that's collecting data over the Sound right now. It's an autonomous system that sends a short pulse of green light into the water. And we record the copolarized and cross-polarized return as a function of depth to find thin plankton layers in the Sound. These layers develop on density gradients and are important to the ecology at the base of the food chain. The laser system itself is autonomous, flies in a small 4-seat aircraft, and collects data. We also can download the data to the hotel here in real time using the antenna to my left and see what's going on. When we find layers in the Sound, we'll call the boat that we're working with and send them out to take samples and measure the density profiles to get more detailed information than we're able to get from the laser. If you're going from place to place sampling with a boat and see a change, you're never sure whether it's changed because you've moved from one place to the next, or it's changed over time as you're doing the transit between it. The aircraft provides an almost instantaneous snapshot of the whole Sound. We're doing 2 flights a day in the Sound here. We'll do an hour to hour and a half flight in the morning and then go out to the airport and grab the laptop that has all the data in it, bring it back to the hotel, take a look, get some lunch, and adjust the afternoon flight pattern based on what we see in the morning. At the same time, we'll call the ship and tell them what we're seeing where so they can go out and sample interesting places. This is about 3 minutes worth of data from the airplane. We're looking at the copolarized and cross-polarized returns shot by shot. Here we have a map of East Sound and West Sound. The black line is the track that the airplane is going to follow, and this is a depth range with intensity of the return recorded in grayscale. This dark region here is the bottom. This is a reef coming up, and the plankton layer are these more diffuse regions that start along here, go along there, and there's another one that starts about this shallow area here and comes up toward this reef. These data are being used for 2 purposes. One is further development of the Lidar technology for ecosystem research, and the other is to study the dynamics of these plankton blooms, their evolution and decay, and how that depends on the structure within the water column. One of the reasons for trying to understand this is because harmful algal blooms are becoming more prevalent and more widespread, and we're trying to understand why that is. The particular bloom that's going on right now in the Sound is a species called Noctiluca, which is in fact creates a toxic bloom in sufficient numbers. And these thin layers do concentrate those, the thin layers do concentrate the plankton and create conditions that are more conducive to a harmful algal bloom.